Have you thought to yourself, so many codons, so little time? Well, do not worry for a second, because this video has been designed to review and simplify the most testable concepts for testing. My name is Brad Ackerson, and this is the MCAT Science Review video on the genetic code. We will start with the bread and butter of what the genetic code does, the central dogma. After, we will review three types of RNA: messenger, transfer, and ribosomal RNA, and we will finish with a look at the codons, which make it all work, and mutations, which make it all not work. Our learning goal for this video is to distinguish between the different types of RNA in our agenda and the different types of genetic mutations. Our learning goal will enable us to answer MCAT-style questions like this one about mutations and their effects on an encoded peptide. So let's get going. We will begin with the central dogma of molecular biology, but first let's define some important vocabulary to lay a foundation. DNA undergoes replication in order to make more DNA. To become mRNA, DNA undergoes transcription. RNA can also undergo reverse transcription and become DNA. In order to make proteins, RNA is translated. The central dogma simply describes this process of DNA becoming mRNA through transcription, and through translation becoming a functional protein. So how does this work? First, transcription. The coding strand contains complementary codons to the template strand, which is why it looks identical to the finished mRNA, with the exception of T in the DNA and U in the RNA. Now, here comes the confusing part. Make sure you keep this straight. The template strand is read in the 3 to 5 prime direction. The mRNA then is synthesized in the 5 to 3 prime direction, making it complementary and anti-parallel to the template strand. The ribosome then reads the mRNA in the 5 to 3 prime direction and synthesizes the protein from the amino terminus, or N-terminal, to the carboxy terminus, or C-terminal. Note that transcription takes place in the nucleus, while translation occurs in the cytoplasm. So how do we get from DNA to mRNA? The RNA polymerase makes it all happen. This enzyme is in the nucleus of the cell and transcribes the template strand. Let's take a look at this segment of mRNA up close. mRNA are broken down into 3 nucleotide segments called codons. How these codons lead to proteins is a little bit different for eukaryotes and prokaryotes. Eukaryotic mRNA is monocistronic, meaning that it codes for only one protein. For example, in a eukaryotic cell, this protein would be protein A. Prokaryotes, on the other hand, are polycistronic, and one mRNA can lead to different proteins. The translated protein depends on where it begins on the mRNA. For example, the same protein A could be translated starting at the beginning. Starting in the middle, however, can yield an entirely different protein, protein B. Our next type of RNA is the transfer RNA, or tRNA. Here we see an example. Each tRNA contains a folded strand of RNA that includes a 3-nucleotide anticodon seen here. The ribosomes are responsible for grabbing the tRNA and pairing it with the correct codon on the mRNA. In order for the process to work, the tRNA must be paired with a specific amino acid. At the 3' end, the adenine has an OH bonded to it. In order to attach the amino acid, the energy from ATP will be used by the enzyme aminoacyl tRNA synthetase. It attaches the amino acid to the 3' end, which always ends in CCA. Once the amino acid is added to the tRNA, it is said to be charged or activated. The activated tRNA and mRNA strands are both grabbed by the ribosome for translation. Ribosomal RNA, or rRNA, is synthesized in the nucleolus and is integral to the function of ribosomes. Some ribosomal RNA act as enzymes called ribozymes. Ribosomal RNA reads the mRNA, grabs the correct tRNA, and catalyzes the formation of peptide bonds. We have thrown this word codon around a few times so far. What is a codon and how is it important to the central dogma? As mentioned, the codon is a 3-nucleotide sequence that codes for amino acids. This can be a bit tricky, so make sure you keep this straight. Each codon only codes for one amino acid. However, each amino acid can be represented by multiple codons. Let's take a look at this figure to reinforce this information. First, what amino acid is coded for by the codon GCG? To find this codon, we go here in the table, to the G row and C column, where we find that GCG codes for alanine. Here we see that one codon codes for only one amino acid. Now, which codon codes for alanine? Here we see GCU, GCC, GCA, and GCG. What we just realized is the definition of degeneracy, multiple options for one amino acid. Let's use isoleucine for our next example. How will the tRNA recognize the codons? The tRNA that supplies the amino acid will be anti-parallel. In the case of AUC, we can know which tRNA will bring isoleucine by taking the anti-parallel codon, in this case UAG in the 3 to 5' direction. Now there are only 4 codons that you have to memorize. They are the start and stop codons. There is only one start codon, AUG, which codes for methionine. When a ribosome reaches an AUG, it begins the process of translation. The 3 stop codons are UAA, UAG, and UGA. How on earth are we going to remember these? Well, for the stop codons, think of this clever mnemonic. You are annoying, you are gone, you go away. For the most part, this process we've discussed is flawless. However, problems can arise, and we call these problems mutations. First, let's define the wobble position. In the reading frame, the wobble position is the last nucleotide in a codon. As we saw with alanine, this can change, and yet the coded amino acid will be the same. When one amino acid changes, we call it a point mutation. Look here, for example, at this mRNA. If we change the A to a G, it still codes for leucine. This is called a silent mutation, as there is no change in the amino acid sequence. Next are the missense mutations. If the T is changed to a C, the beginning stays the same, but the leucine is switched for a serine, resulting in one new amino acid in the sequence. Nonsense mutations occur when a point mutation results in a premature stop codon. Again, the beginning is fine, but the stop codon terminates translation, leading to a shorter and generally non-functional protein. Frameshift mutations occur when a nucleotide is added or deleted from the mRNA. If we remove the T, for example, everything upstream of the T is fine, but everything after that has changed. This is bad news, as the protein will not serve its original function, and many frameshift mutations lead to one of the premature stop codons eventually. Make sure you understand the differences between these types of mutations. Let's see how the MCAT will test these concepts. Answer this first question about the different types of mutations. The correct answer is D. If needed, review the mutations segment of this video until you feel comfortable defining each type. Answer this next question on linking an mRNA sequence to its corresponding amino acids. If you chose D, you are correct. Attack these questions one amino acid at a time. For example, serine must be coded for by one of these 6 codons. Comparing that to the answer choices, we can eliminate A and C. Next, we go to alanine, which must be coded for by one of these 4, excluding B, and D is correct. Let's review the main things to learn from this video. For the central dogma, remember that DNA is transcribed into RNA and translated into proteins. mRNA is used as a template for translation, transfer RNA carries the amino acids, and ribosomal RNA is a major component of the ribosomes. There are 4 important codons that you need to remember for test day. They are the start codon, AUG, and the 3 stop codons with their accompanying mnemonic. You are annoying, you are gone, you go away. Do not memorize any other codons. And last, the different types of mutation. Silence, missense, and nonsense are all single base change mutations and result in varying effects. The frameshift mutation results from the addition or subtraction of a base and drastically alters the polypeptide protein sequence. Thank you for watching, and please feel free to review this video as much as needed to feel confident about these concepts on test day. Best of luck!